Dear students, today I will describe the histological structure of the liver and the pancreas. If you have a look at the microscopic slide on the liver, it looks chaotic at first. You can see a couple of holes and randomly chaotically distributed epithelial-like cells. To better understanding the structure of the human liver, we show you also in the histological practice a pig liver. The difference between these two liver types is that the pig liver has very well visible connective tissue septa between the units of the liver, what we named lobule. By the way, beside pig, there is only one more animal, a known animal, in which this type of connective tissue septa is the polar bear. These units, these hexagonal units, this is the classic units of the liver named lobule. In the middle of the lobule we have a vein, the so-called central vein, and around that we have the liver cells. In the corner we also have structures, and because we have three of them, three different type of structures, and all of them are connected somehow to the porta hepatis, they named portal triad. The members of the portal triad is the interlobular artery, the interlobular vein, and a bile duct named the ductus biliferus. By the way, you can see the veins in the liver has no practical wall, no wall other than the endothelial cells and the basement membrane, since the liver is hard enough to maintain the integrity of a vein. Between the liver cells, we have liver sinusoids, which are modified capillaries. The structure will be described in detail later on. And the liver cells are organized usually more or less vertically integrated laminas. The blood circulation of the liver lobule is that from the corner, from the portal triad, the blood flows inside to the center through the liver sinusoids, then for the central vein to the sublubar vein is away. This was a kind of general introduction to the structure of a liver. Now let's see some details. The main characters of the livers are the liver cells or hepatocytes. If you have a look at that, here these cells are epithelial-like cells, and interestingly many of the cells has two nuclei, like here or so. Even if the cell has only a single nucleus, it has many couples of genetic material from 2 to 8 in a cell. The reason for that is the liver cell lives a very active life, and it must synthesize a lot of proteins. And for protein synthesis necessary a very speedy system. As you know, for most of the genes we have only one copy in a genome, and it takes time until new messenger RNA copied and so on and so on. Just to speed up this process, we need multiple number of genes, and because the genes themselves cannot be duplicated, cannot be multiplied, the entire genome is multiplied. This is why these cells are polyploid. Inside the cells, we have a granulated endoplasmatic reticulum, we have smooth endoplasmatic reticulum, many free ribosomes, also mitochondria, we have glycogen and fat particles, and also we have liposomes. You know, it's aggressive enzymes in the membrane compartment, which is necessary for digestion, some of the stuff. Between the cells, we have the bile capillaries. The bile capillaries has no own wall. It can be considered as if it were a gland, a tubular gland, an lumen of a tubular gland. In an electron microscopic interpretation, you can see that we have a microvilli which increasing the surface, so it's much easier the bile secretion, and it has a very tight connection, zonal occludence and adherence type of connections, just to prevent the leakage of the bile to the outside world. The intercellular space is interesting. The surface of the cells has microvilli, and whatever spaces between the cells, named the disses space, is a traditional name. The biggest character in the intercellular space are the sinusoids. Sinusoids are modified capillaries with a very transparent wall. It has fenestrated endotheliums, big windows in the endotheliums, so big molecules can pass through it. And also it has no basement membrane. Instead, we just have reticular fibers which keep the endothelial cells together. The reason for this transparency is that large molecules must pass normally through that. As you know, the liver produces most of the blood proteins. Blood proteins have high molecular weight, so it wouldn't pass through the basement membrane if it were there. And also, one of the functions of the liver, as you saw in the anatomy lecture, is to finalize the digestion. So big molecules must be picked up, pass through the capillary wall to get further digestion. In the sinusoids, we have Kupffer cells. This is the electron microscopic interpretation, elongated cells anchored to the wall. And you can very nicely see them also in the light microscopic slides as elongated cells inside the sinusoids. These are macrophages. Whatever particles are in the liver sinusoids, they will eat it up. Very notable particles are the fragment of the erythrocyte membrane, which are broken in the spleen, and through the portal circulation they led to the liver. The other typical cells are the E2 cells. E2 cells are between the sinusoids and the liver cells, here represented by this cyan-colored cell. This has very complicated morphology. They have large lipid droplets in the cytoplasm containing high level of vitamin A. And practically these cells can be considered as modified pericytes. These are practically stem cells, so can be differentiated in any other type of cells, and they play a very important role in the regeneration of the liver. As you know, if you have your liver damaged mechanically, even if you lose 2/3 of the liver, in a couple of years, it completely will be regenerated, provided the leftover liver is healthy. The circulation of the liver, it has an artery, they name propria hepatic artery, which is a branch of the celiac trunk and the common hepatic artery. It goes into the lobes, usually in more branches, and inside the lobes they divide into more and more. These branches are named the intralobar branches, and this is one example of them, a large artery there. It's always accompanied by a vein and a bile duct, similar to the portal triad. The next branch is the interlobular artery. I already mentioned this is the member of the portal triad. Then the blood from the interlobular artery will be distributed all around the liver lobule. This is what we name the circumlobular arteries. And finally, the blood from the circumlobular artery go to the liver sinusoids. Interestingly enough, as I mentioned in details in the anatomy lecture, there is another vessel through which the blood goes into the liver, and this is the portal vein, the vena portae. The route inside the liver is very similar to that of the arteries. It has lobal branches, intralobal branches. This big guy is an interlobar vein. Then we have the interlobular vein. Similarly, we have circumlobular veins, and from the circumlobular veins, the blood goes into the sinusoids. Once again, in this system, the blood goes into and not out of the liver. In the sinusoids, the blood mixed from the arteric and the venous blood mixed at the peripheral end and goes centrally. At the periphery, it's already a mixed blood, and there is an interesting pathological consequences of this type of standard blood flow. The liver cell can be damaged for two main group of reasons. Number one, a kind of toxins, poisoning, and the other one is doesn't get enough oxygen. If there is a lack of oxygen in the blood circulation, like heart failure and so on, the peripheral cells still get certain level of oxygen, but the inner cells not enough. So they will necrotize. So in a cell, if we have a lack of oxygen, the inner part of the the kidney lobule, hepatic lobule is necrotized. In contrast, if you have toxins, the outer cells get first the toxins, they will damage and they will die, and because they partially used up these toxins, the inner cell will be relatively sane. The venous circulation, from the liver sinusoid, the blood goes to the central vein, and from the central vein to the sublobular vein, which is a connection of the central veins. The sublobular vein can be seen in a slide as a large vein, and this is alone. If this is the intralobal vein, it's always accompanied by an artery and a bile duct. If it's alone and relatively big, this is the sublobular vein. Finally, they go into the hepatic veins. The hepatic vein has no part outside the liver. They always stay inside the liver, and whenever you see into the inferior vena cava, you can see holes on that. These are the hepatic veins, so they do not go out. In contrast, the inferior vena cava become embedded in the liver to pick up the hepatic veins. Now, this was about the circulation. Let's see the bile circulation. As I described earlier in details, the bile capillaries are wall-less tunnels between the hepatocytes. In this picture, we can see a silver impregnated bile capillaries in the liver. From the bile capillaries, the bile capillaries has a dead end centrally, and the bile flows outside to the outer surface. If we have cell lesions, the cell will die, necrotized. The bile can flow in the place of the cell to the sinusoids, and the content of the bile gets there. This is what the most visible consequence is, the yellowish color, which named it jaundice, which shows a severe cell lesion in the liver. Whenever the bile goes to the surface of the lobule, it will be collected by a duct, a very thin wall duct, parallel running to the circumlobular arteries in the veins, and this is what we named the Herring's canals. Finally, they go to the ductus biliferus. You remember this is one member of the portal triad, and it goes to the bigger ducts. They already have columnar epithelium, and finally the bile goes out to the hepatic duct. The rest was described in the anatomy lecture. Just to tease the students, some books refer some alternative type of unit. Whatever I described is a classic lobule, and this lobule is based on the picture of the pig's liver. So the classic lobule is something like that. We have a central vein in the middle, and in usually three corners of them we have a portal triad, and we have three highlighted classic lobules here. If you see an embryological way, the liver cell develop, as you will see later on in the embryology, as a duct going out from the duodenal loop, and there will be the bile duct system, the Herring's canal, and from that practically the liver cells can be considered as a gland, as a tubular gland, and the secretory part are the liver cells, and the rest of the bile ducts are the duct of this system. So whatever cells, liver cells, belong to a single Herring's canal, which is between the lobules, this area is named the portal acinus. So this is an alternative unit, a kind of embryological or functional unit of the liver. There is another type of unit. If you think that we have a center of the ductus biliferus with the three Herring's canals, all the cells developing from this area, all the cells which are derivative of this bile duct, named the portal lobule, and this is an hexagonal shaped area, and it takes 1/3 of three neighborhood classic lobule. Or the other one, some people think that in every corner we have portal triad. Sometimes it happens. In this case, this is the half side. This is also named in some book as portal lobule. No, I will not complicate the things furthermore. And let's summarize again what are the functions of the liver. As I mentioned in the anatomy lecture, the liver has many functions. It's a common mistake that is primary detoxicating organ. The primary really is a chemical laboratory, and primary task is finishing the digestion, processing the nutritions. What means that whenever the enzymes digest the food, they will not digest into sugar molecules or amino acids, but most of the little groups of sugar or little groups of amino acids, oligosaccharides or oligopeptides. And this is up to the liver to split into units, because only the sugar molecules or the amino acids can be utilized by the other cells of the body. So the portal circulation is serving this purpose. The other function is the storage. It stores the glycogen. So whenever we have excess carbohydrate in the blood, it will be taken up by the liver and stores in the glycogen. And when the cells need sugar, they will release it. Also, it stores a lot of vitamins, especially lipid-soluble, lipophilic vitamins. It produces all the blood proteins, albumins, globulins, and so on. Practically the only major blood protein which is not produced by the liver are the gamma globulins, the immunoglobulins. It produces the bile. Bile has two major functions. It contains the bile acids, which is absolutely necessary for the lipid digestion. It's a very strong detergent just to emulsify the lipids, and also eliminates the lipid-soluble waste. Since we have bile acid there, which emulgates all the fat-containing stuff, it's very obvious that the lipid-soluble waste also can be mixed and can be held into the bile. Detoxification is also happens, but this is not whatever we eating poisons or eating toxins all day long, but mostly internal harmful or unnecessary things. One of the most important functions is turns the hormone content off. When we have a neural control, the nerve cells will send out signals, and whenever they stop sending out signals, actually the action stops. The endocrine system has a little bit different way of working, like producing hormones. They turn the signals off, and that's it. If the signals is circulating in the blood, as long as it's circulating, the signal is active. It's up to the liver to turn it off. Liver excretes enzymes which very quickly eliminates this hormone, and this is how the signal is turned off. Whenever you have severe liver damage, most of the cases, the most important cause of the death is a kind of hormonal chaos. All the hormones are still remain active for a long period of time, and they will be a mess. And this is one of the main reasons what kills the people. Also detoxifies the metabolic end products, especially the hem, whatever is a kind of waste material from the hemoglobin digestion. And of course, we have also sometimes the food. The food industry takes care of for us to eat many different toxins, harmful material in the food. It produces hormones, as I mentioned before, and as you know from other subjects, the IGF-1 is a very powerful and very important hormone family, and angiotensin is one of the main character of the blood pressure control. In the embryonic life, it's also produced. blood. Now that was about the histological structure of the liver. Let's go for the neighborhood thing, the gallbladder. A gallbladder has a relatively simple structure. It's a layered organ, so the inner layer is a tunica mucosa. The epithel is very nice, simple columnar epithel. Maybe this is the most beautiful simple columnar epithel in our body, and it has microvilli on the surface. Below the lamina propria, like always, it's loose connective tissue. In this case, we have a very, very rich fold system. Almost, sometimes it makes a very, very interesting formation. In the lamina propria, we can sometimes see tubes which has similar epithel as in the surface. This is what we name the Ashok-Rocky sinuses. This is a practically outer artifact. So if you imagine that we have this area and we make a cross-section of that, we can see a round closed area. This is something like that. So this you can see a big pouch coming against us. So practically it's no other importance. Sometimes it's very deep, that might have some pathological consequences. The muscular layer made of smooth muscle, and the smooth muscle is practically a network-like. In some areas of the gallbladder we have parallel running fibers, but most of them are bundles running in every direction in the wall. The outer layer is the adventitia of serosa. It's loose connective tissue. On that part of the gallbladder, whether it's covered by peritoneum, this is named the serosa because we have a mesothel on the surface, and whenever it's contact with the liver, just through connective tissue without the peritoneum, this is named the adventitia. In the adventitia, sometimes we also see ducts named Luschka's ducts. These are developmental malformation. This is actually the siblings of the liver, the gallbladder, and whenever you study the development of gallbladder, you will understand that. Now just you have to remember this name. Finally, I will talk about another interesting organ, the pancreas. The pancreas is a complex organ, a complex gland, which has an exocrine and endocrine components. The exocrine components consist of serous gland-like structures, which looks pretty much like serous glands, a round group of cuboidal cells, which mainly, basically, blue cytoplasm. However, it has a difference between the standard serous gland and this one. This is just in contrast, it's a parotid gland, and this is the pancreas. You can see the serous gland, the parotid gland has homogeneously blue cytoplasm, unlike this one, which blue at the periphery and red close to the midline. So it has a double color. The reason for that is that the ribosomes, which are responsible for the blue color of the parotid gland, more or less evenly distributed in the cytoplasm. In the pancreas, is differentiated mostly in the basic. The ribosomes are sitting, this is why the base is blue, and inside you already have a ready-made secretory granules, the enzymes, which are proteins, and this is what makes the tip of the cells red. The other difference is we have white cells, whitish cells in the middle. This is what we name the centroacinar cells. The centroacinar cells can be considered as a push-in duct. Probably they have some protection against the enzymes these cells produce. This is a kind of structural picture on that. These blue cells are the cells which are the duct, and apparently as if it were pushed into the lumen, very probably to protect the cells. If you see the other structure, you can very nicely see all of that what I mentioned before. On the base we have large quantities of rough surface endoplasmatic reticulum, a lot of ribosomes. This is why the base of the cell is blue. And in a tip we already have zymogenic granules, secretory granules, and which contains the ready-made enzymes. The role of the pancreas is to produce enzymes. It produces a large quantity, practically every type of the digestive enzymes. When you will go to exam in biochemistry, you have to know at least 30 different enzymes. In physiology, about 10. In anatomy, at least 2-3 you have to mention. And the trypsin may be the best known pancreatic enzyme. So this was the endocrine part. Exocrine, but endocrine part is present in islands, named the Langerhans islet, and the structure is different. We have approximately 100 to 200 micrometer in diameter. It's almost visible with naked eyes, and we have a variable number, 200,000 to almost 2 million. Of course, the size is different, so the total mass is not that different from human to human. The cells are located not in rings, not in acini, but cords, and the cells are pale. This is even paler than the average cell because the hormones, the insulin and the glucagon, can be dissolved during the slide production. The cord-like position of the cells is due to the fact that they are endocrine cells. The endocrine cells are emptying their product into the capillaries, and they must line up along the capillaries, and this is why we have longitudinal cords, longitudinal group of cells. We have an interesting flat cells which separates the endocrine from the exocrine. This is shown yellow in this picture, and based on genetic studies, they're supposed to be the Schwann cell type of cells. This can be first very surprising, but if you think about that, during the development, the neural crest, whatever cells coming out of the neuroectoderm but not involved in the neutral tube, will develop any kind of control substances in the periphery. Peripheral nerve cells, peripheral sensory cells, peripheral endocrine cells, and also the service cells. Like Schwann cells is a service cell for the neurons. It means making the myelin sheath, and here is also making a kind of service separating the endocrine and the exocrine cells from each other. What type of cells we are? We have 3 types of cells. The alpha cells, which are a kind of minority, about 20% of the cells, they're producing glucagon and mostly located in the periphery. In the hematoxylin eosin stain slide, it's impossible to distinguish these type of cells, so you are not requested to distinguish them. Whatever, you have to know that it's mostly located close to the peripheral area of the Langerhans islets. The glucagon is a peptide hormone which acts on the liver, and it initiates liberating glucose from the glycogen. And this is this task. Whenever the blood sugar lowers down, they produce glucagon and initiates the liver to send glucose into the blood. The insulin, which makes the majority of the cells, loosely located in the middle of that, they produce insulin. The target of the insulin is every cell, because insulin is required for the cells to pick up the glucose. Glucose is absolutely necessary component of every cell, because this not only a kind of metabolic part, but also it's a fuel, it's an energy source of that. Without insulin, most of the cells, with a few exceptions, cannot pick up glucose. Consequently, the insulin is absolutely necessary for the cells to pick up. It's very important to dismiss a kind of misunderstanding. Primitive lecturer says that the alpha and the beta cells are antagonistic because the alpha cells increase the blood sugar and the beta cells decrease, like if they were playing with a Libicoke. No, they are working together and doing 2 consecutive steps of the energy supply of the cell. The alpha cells will send from the liver to the blood the glucose, and the insulin from the blood to the cells, so they are working together to energize, to feed the cells. The third side of the cells are the delta cells or D cells. Actually, there was a gamma cells, but it was turned out during the history of the sciences. It was a not real one, it was a cheat. And this is why the real third type of cells, named the delta cells, the fourth letter of the Greek alphabet, also D cells. And these are producing GI hormones, especially somatostatin and VIP. As you know, that this contrast is that they are hormones are paracrine hormones. They acting in a neighborhood, they never produced in such a high quantity that it would be exposed to all over the body. Consequently, this is also just controlling the neighborhood alpha and the beta cells, somehow modifying the things. We have special staining to distinguish especially the alpha and the beta cells. A traditional staining method introduced about 50 years ago was the aldehyde fuchsin by Gomery, and these violet color cells are beta cells only, the rest of them are different. And now the sure detection, the modern detections are the immunohistochemistry. Here you can see glucagon. This is immunohistochemistry, antibody against glucagon. Consequently, these dark brown cells are the alpha cells, and this was made by antibody against insulin. And you can see that the insulin-containing beta cells mostly located in the middle of that. The other structure you can see, you can compare the exocrine and the endocrine cell. Exocrine cell has very big secretory granules which contains enzymes, as you remember. The endocrine cells has small secretory granules, so there's just a slight morphological differences between them. And that's what I wanted to talk about, the histology of the liver and the pancreas. Thank you very much for your attention.